Okay, part 2 of question 5 then. The figure shows a representation of a short polypeptide. You should know that that term is to do with proteins. A polypeptide is a sequence of amino acids, okay, which codes for a protein. So polypeptide chain made from 3 amino acids. So here we have our 3 amino acids. Don't worry about the names and so on. They're joined by a specific bond, which we'll come to in a second. And here is your basic structure of an amino acid. We draw it like this. You can do it the way that it was in that first question, that's fine too. But this is the way we've sort of learnt it. Because if we start with our oxygen in the top right, that's how we draw a lot of our other molecules. It just keeps it a bit more simple. So we've got NCC. Remember when I got that Eminem song wrong? Our oxygen here, double bond because it needs 2 and it's just bonded to the carbon. Hydroxyl group, R group here, and then the rest are hydrogens. And the R group we know can consist of any of these molecules and could also be sulfur, remember. Okay. So the question says, name bond W and state what type of reaction takes place to form this bond. So again, use this as an opportunity. If you can't remember your bonds, make a flashcard, postcard, blah, blah, blah. So in lipids, the bond between the glycerol and the fatty acids is an ester bond. In carbs, holding the sugars together is a glycosidic bond. That's the first ones we learnt about, glycosidic bond. And in proteins, holding the amino acids together, it is a peptide bond, polypeptide, peptide bond. Okay, so you just got to know these ones. And all of these bonds, all are formed by condensation reactions. Which means you put H2O, hold on, no it doesn't. It means that you get, H2O coming off of it. And then if you hydrolyze those bonds again, that's a hydrolysis reaction, you add the H2O back in to break it up again. So name the bond, name the bond and state what type of reaction takes place to form this bond. Well, it's an amino acid, which is proteins joining the amino acids together. So it's a peptide bond. And like with all the others, it's a condensation reaction. Again, a straightforward one if you know your bonds. If you don't, go and learn these. The R group is in the amino acid alanine is CH3. So we can look back here. We know that the R group here is CH3. So let's just draw that out to help us. NCC, oxygen, not carboxyl, hydroxyl group. R group is CH3, or you could draw it as a C with your 3 H's coming off. Then our hydrogens. Okay, describe the structure of the amino acid alanine. Right, it's 4 marks. So we've got an amino group or amine group at one end. So point number 1, there is an amino group at one end and a carboxyl group carboxyl group at the other end. Okay. The central carbon, or the carbon in the middle, has a single hydrogen attached to it. And the R group is CH3. That should get you all the marks. So amino group, 1 mark, carboxyl group, 1 mark. Central carbon has CH3. I'm just trying to think back to the mark scheme. I think you get quite a few marks just for drawing it out as well. Another thing you could say, and I will put the mark schemes up for you by the way, another thing you could say is that CH3 is a methyl group, okay, which is just an extra thing which you should know from the structure of fatty acids, triglycerides and so on, that CH3 is a methyl group. It's just called a methyl group, like a hydroxyl carboxylic group and all of that, okay? So CH3 is a methyl group. Okay, put that there if you wanted to pause, and I'll stop it there.